Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing IISER Aptitude Test IAT conducted in the year 2021. I will be doing only the biology section. If you are new to my channel, please do like, share and subscribe. Coming to the first question. Which one of the following epithelial cell types is commonly found in the inner surface of the fallopian tubules? So it is the ciliated epithelium that is found in the inner surface of the fallopian tubules. So answer is option A. Then next one. Which of the following is not an asexual reproductive structure? Isogametes of cladophora. Isogametes of cladophora, they are sexually reproducing structures. So that is not an asexual reproductive structure. So option A is the answer. Whereas conidia, zoospores, gemmules, all these are asexual reproductive structures. Then next one. Which one of the following is an example of palindromic DNA sequence? Palindrome means it should be read in the same in both the direction. That is, if you take the case of DNA, it should read the same in 5' 3' direction in both the strands. The direction should always be 5' to 3'. So if you see option A, you can see it is GAATTC in the 5' 3' direction in the top strand, and then in the bottom strand, if you read in the 5' 3' direction, it is again GAATTC. So option A is a palindrome. Whereas others, if you see, it is GACTTC. Then again, if you read in the bottom strand, GAAG, it is nowhere matching to GAC. So it is not a palindrome. See option C, it is GAA. Then here, if you see, it is GAC. So it is not a palindrome. Again in D, it is GAC and CTG. So again, it is not a palindrome. So the answer here is option A. Then next question. Some individuals start sneezing when the pollen content is high in the air. Primarily, which Ig isotype will these individuals produce as an immune response? So they have asked Ig isotype. So whether it is G or A or M or E or D. So in this condition, that is sneezing when pollen content is high in the air, it results in IgE response in the individual. So answer here is option A. Then next one. Which of these factors is likely to cause least likely to cause deviation from Hardy-Weinberg equilibrium? So in order for the population to be in Hardy-Weinberg equilibrium or under non-evolving condition, then it must meet 5 major assumptions. So which are they? There shouldn't be mutation, there should be random mating, there shouldn't be gene flow, the population size should be very large, and no natural selection should occur. So if these conditions are met, then the population is in Hardy-Weinberg equilibrium. So here they have asked which is least likely to cause deviation. So if there is mutation, gene flow and genetic drift, then it will cause deviation from the Hardy-Weinberg equilibrium. But if there is a small reduction in population size or reduction in population size, then it is least likely to cause deviation from the Hardy-Weinberg equilibrium. So answer here is option A. Then next one. Motivated by the classical experiment of Frederick Griffith and the work of Avery, MacLeod and McCarthy. So Frederick Griffith was the one who found the transforming principle, and Avery, MacLeod and McCarthy, they conducted again the experiment and found that the transforming principle is nothing but the DNA. To identify the transforming principle, a scientist redesigned and performed the experiment with S strain and R strain of Streptococcus pneumoniae as summarized in the table below. So this is the Griffith experiment, just for your revision and easy answering of these questions. So this is the rough strain. It is non-virulent. The mouse lives upon injection of the rough strain to mouse. Now if it is the smooth strain or the S strain, the rough strain is R strain, smooth strain is S strain, and S strain is virulent and the mouse will die. Now at the same time, if you use heat killed S strain, the mouse will live. If you use rough strain and heat killed S strain, then DNA from heat killed S strain will be transformed to the rough strain and the mouse will get infected with the disease and the mouse die. So this is the Griffith experiment. Now we will come to the question. So this was the treatment and these are the experimental conditions which the scientist redesigned again as per the Griffith experiment. So T1 is from the options below, identify the outcomes of the treatment T1 to T6 on the viability of the mice. So T1, S strain heat killed injected into mice. So the mice will live because S strain is heat killed. Now T2, S strain heat killed plus R strain live together injected into mice. Again the mouse will die because S strain is heat killed but it has transformed its DNA to the R strain. So the mouse will die. Then next one is nucleic acids isolated from S strain heat killed plus R strain live incubated with RNAs and injected into mice. The mouse will die because sorry. So S strain heat killed and R strain is live. Heat killed S strain would have released its DNA which has already transformed the R strain. It was incubated with RNAs, so RNA only will be degraded but DNA will not be degraded. So the mouse will die. Then the next one, nucleic acid isolated from S strain heat killed plus R strain live incubated with DNA. So DNA means it will destroy your DNA. So R strain is not going to be transformed with S strain DNA and inject into mice means the mouse will live. T4 live. T5 S strain live injected into mice, it will die. And T6 R strain live injected into mice, it will survive. So your answer here is option A. Then next one. Which of the following is produced by a plant during photosynthesis with far red light? So cyclic phosphorylation is there and non-cyclic phosphorylation is there, and these two they will produce ATP and NADPH. So both ATP, NADPH and H plus will be produced. Answer is option B. The next one. The map of a 4000 base pair plasmid marking the location of different restriction enzyme cut sites is shown in the figure below. The numbers in the brackets indicate the base pair positions where the enzymes cut. This plasmid is completely digested first with the combination of restriction enzymes PST1 and HIND3 and then with the EcoR1 and SAL1. So the plasmid, which is 4000 base pair in size, is first digested with PST1 and HIND3. Then again after digestion with PST1 HIND3 you add EcoR1 and SAL1 to it and then digest it. The final digested plasmid is analyzed by agarose gel electrophoresis. So this is the plasmid. This is PST1, EcoR1, HIN3, SAL1. This is 800 base pairs from the origin, 1700 base pair again from the origin, 1920 base pair from origin and 2460 base pair from the origin. This is the 4000 base pair plasmid. So which one of the following option correctly represent the band size in base pair obtained on the gel? So this is the plasmid which is digested first with PST1 and HIND3. First it is digested with PST1 and HIN3. So you will get PST1, HIN3, one side, one band plus this vector backbone or the remaining part of the vector or the remaining part of the circle. That would be much easier. Instead of telling PST1 eco R1 HIN3, I will say circle. Then you will understand it very easily. So, SAL1. Okay. So, this is PST1 and HIN3 digestion. Next, you digested this product with eco R1 plus SAL1. So, what do you get? You get PST1 eco R1. This one, because HIN3 portion is being cut off. And eco R1 HIN3, this small portion. Then HIN3 SAL1 portion, this portion you will get, and you will get the vector backbone, this one. Or the remaining part of the circle after cutting. Okay. Now we will see the products of digestion. So first eco R1 HIN3. I will take this portion, eco R1 HIN3, eco R1 HIN3. That will be eco R1 will be HIN3 is I forgot to add the base pair here. HIN3 is 1920 and eco R1 is 1700. So this portion will be now 1920 minus 1700. This will be the size of this particular digested product. This one, eco R1 HIN3. How much is it? It is 220 base pairs. Then comes HIN3 and SAL1. This portion will be SAL1 is at 2460 and HIN3 is at 1920. So you subtract it and you will get 540 base pair. That is, this region will be 540 base pairs. Then eco R1 PST1. Eco R1 PST1. This after removing of this particular region, this band will be remaining there. This band, this portion. So this portion will be remaining. That will be PST1 eco R1. So it will be 1700 minus 800. So it will be 900. So it will be 900 base pair. PST1 eco R1. HIN3 SAL1 eco R1 HIN3. So all three we got. Now we have to see what is the size of the remaining vector backbone or remaining part of the circle, how much it is. For that you have to know the size of PST1 HIN3, this portion, because this portion is being digested, cut into pieces, PST1 eco R1 HIN3. So this total how much it will be. So PST1 to HIN3, that will be PST1 is at 800 and HIN3 is at 1920. So 1920 minus 800 will give you 1120. Then SAL1, this portion, HIN3 SAL1, this portion was also digested from the plasmid. So how much it will be? It will be 540. We already got it over here. How much is this portion? We got it as 540. Now add up 1120 plus 540. You will get it as 1660. That is region digested and removed from the 4000 base pair plasmid. That is from this size. This region has been removed, cut off and removed from the plasmid. Now the remaining, that is this portion will be 4000 minus 1660, that is 2340 base pair. So your answer here will be option A, that is 220, 540, 900, 2340. 40 base pair. So the answer is option A. Then next one. The following plot represents changes in the rate of photosynthesis with leaf temperature for two plants P1 and P2. This is P1, this is P2, this is rate of photosynthesis and this is the temperature. Which one of the following statements correctly describe the characteristics of P1 and P2 plant? So P1, if you see, it is having lower temperature optimum. So it is going to be a C3 plant. Whereas P2, if you see, it is having a higher temperature optimum. Like this. It is peaking at 20. This is over 35 to between 30 and 40 or 35, nearly 35. So it is C4 plant. And C4 plant, they respond to higher temperatures and show higher rate of photosynthesis. They are tropical plants. And C3 plants, they have lower temperature optimum and they are temperate plants. That is, they have temperate adaptation. So the answer here is option A. That is, P1 is a C3 plant having temperate adaptation, whereas P2 is a C4 plant with tropical adaptation. Answer is option A. Then next one. The figure represents two categories of animals, X and Y, with respect to their response to the external environment. This one, their internal body temperature and external temperature, X and Y. Figure 2 represents two broad categories of animals, type A, type B, with respect to their body surface to volume ratio. So surface area to volume ratio and type A, type B animals. Which combination most suitably represent the following animals in the order? Hummingbird, crocodile, frog, polar bear. Now before answering this question, what does this graph represent? This one is X and this line is Y. So X is showing a constant body temperature independent of the external temperature. So X is likely to be an endothermic animal. Whereas this is Y, it is an ectotherm because the body temperature changes with temperature of the environment. That is, internal body temperature changes with external temperature in the case of Y, whereas it is almost constant or it is constant in the case of X. So Y is an ectotherm. Now coming to this. So here type A, if you see, it is having a higher surface area to volume ratio, and type B, it is having a lower surface area to volume ratio. Now we will see the animals in their order. First is hummingbird. So hummingbird, they are small sized animals. They have larger surface area to volume ratio. So it is type A. That is type A. And what is it? Is it an exotherm or an endotherm? It is an endotherm because birds and mammals, they are endothermic. So XA. XA. Then comes crocodile. So crocodile has got larger body size. So it has lower body surface area to volume ratio. So it belongs to type B. Type B, and crocodiles are ectothermic animals, hence YB. Then comes frog. Frog is having smaller size. So it has high body surface area to volume ratio. So it is type A. And it is an ectothermic. So it is Y. It is a reptile. Sorry, it is an amphibian. So it is YA. Then comes polar bear. Polar bear, larger size. So low surface area, body surface area to volume ratio. So it is type B, and polar bear is a mammal. It is an endothermic. So XB. So the answer here is XA, YB, YA, XB. Answer is option A. Then next one. In plants, ammonium ions are produced by protonation of ammonia. Which enzyme uses these ammonium ions to convert an alpha keto acid into an amino acid? It is glutamate dehydrogenase. Nitrogenase, it is involved in nitrogen fixation, so it doesn't have anything to do with amino acids. Trans-acetylase, it is involved in transferring acetyl groups, not amino groups. So C is also not correct. Lactate dehydrogenase, it converts pyruvate to lactic acid, so it is not involved in amino acid synthesis. So the answer here is option A. Then which one of the following physiological functions is common between the small intestine and the renal tubule? It is absorption of glucose. Because glucose is a common nutrient that is absorbed in the small intestine and reabsorbed by the kidney tubules. Almost all glucose is reabsorbed in the proximal convoluted tubule part of the nephron. So answer here is option A. Then next one. Match the following pairs of interacting species to the corresponding names of the interaction. Herbivores and plants. Herbivores and plants can be considered as predators. Herbivores predate on plants. They eat the plants. Then so one will be B. 1B. Okay, 1B. Then cuckoo and crow. Cuckoo and crow is pakka parasitism. So parasitic relationship. 2C. Cuckoo will put its egg in the crow's nest. So 2 will be C. Then sea anemone and clownfish, it is commensalism. The sea anemone is neither harmed nor benefited. So it is commensalism where one species is neither harmed and benefited. So 3D. 3 will be D. Fungus cyanobacteria, that is lichens, it is mutualism. 4A. So 1B, 2C, 3D and 4A. So answer here option A. Then next one. The following events are associated with meiosis. Which of the following is the correct sequence of these events during meiosis? Now before coming to that, what are the stages of prophase 1 in sequence? Leptotene, zygotene, packetine, diplotene and diakinesis. Now first one will be formation of synaptonemal complex. The first and that is occurring in the zygotene stage. They haven't mentioned anything about the leptotene stage, but zygotene, packetine and diplotene have been mentioned in the statements that I will explain. Formation of synaptonemal complex, that is in the zygotene stage. Synaptonemal complex that holds homologous chromosomes together. So that's happening in the zygotene stage. So this one will be the first one, that is 4 will be first one. Then comes formation of recombination nodules. Recombination nodules are the site at which non-sister chromatids cross over. Formation of recombination nodule is characteristic of the packetine stage. So after zygotene you have got packetine, that is 2. Then comes chiasmata. Formation of chiasmata. Chiasmata become visible during the diplotene stage. So zygotene, packetine and this is diplotene. So it is formed due to the dissolution of synaptonemal complex and separation of homologous chromosome except at the sites of crossover. Then finally is the formation of meiotic spindle. Meiotic spindle is for the separation of homologous chromosomes of the non-sister chromatids. So the answer here is option A, that is 4, 1, 3 and 2. Answer is option A. Then next one. Which of the following floral formulae represents a zygomorphic flower with a diadelphous androecium and superior ovary? So as per me it is option A, because that is having 9+1 arrangement where 9 anthers are united in filament and one is left free. G1 with the line below the G is showing it is superior ovary. So I will explain all of these. What does this mean? This percentage symbol, it means the symmetry of the flower is zygomorphic. This symbol means it is bisexual flower. K5 and 5 means 5 K indicates calyx and 5 sepals are there, and in brackets they show that they are united with each other. Then this is corolla. Corolla is the number is 24, 5. So 5 petals are there, and it shows vexillary estivation, that is 1 plus 2 plus 2 in brackets, that is vexillary estivation, and it is characteristic of the family Fabaceae. Then A is 9 plus 1. This means andrecium. A means andrecium, and number 9 plus 1 represent diadelphous stamens. There are total 10 stamens. That is 9 plus 1 is 10 stamens, and 9 in brackets means the filaments are united together to constitute one bundle, and one here, this one, it means one filament is left free. So it is a diadelphous andrecium. And G1 with a line below G, it shows it is superior ovary. That is line below G, it represents superior ovary, and the one over here represents monocarpellary ovary. So the answer here is option A. So that's it. Thank you for watching my channel. Please do like, share and subscribe. Thank you.